Hello, my dear friends. Welcome to Live Prep. Myself, Dr. Gajapati Verma. As I informed you earlier, under the name of Bootcamp, we are providing revision lectures and also we are conducting test series. These revision lectures are available both on Live Prep app and also on our YouTube channel. But test series can be given only through the app. So now in this particular video, we are going through the revision of the chapter that is Biotechnology Principles and Process. The entire chapter we are covering in single video. And here please remember, we are confining ourselves to NCERT content only. That is one point. Second important point is, I am not exactly going through NCERT sequence, the same content according to our convenience. So I am keeping them under different headings so that in a particular flow we can cover that particular content. But entire content of NCERT we will try to cover here. Clear? And now let us quickly go through this. This is the paragraph that was given under the unit introduction, and here also there are some important sentences are there, so you can go through this. The whole approach to understand this natural phenomena became anthropocentric. What is the meaning of anthropocentric? So anthropo means a human at the center. That means for all this knowledge, so human based development is taking place. So in that sense actually they are talking here. Next one, the major utility of the biological world is as a source of food. So in the major cases, so with respect to the human, if you are talking, so majority of the other organisms, other things, so we are actually talking with respect to the food source. So that's what they are actually talking here. Biotechnology, this is the 20th century offshoot of modern biology. Please observe carefully. These sentences you need to remember as it is. So the 20th century offshoot of modern biology is biotechnology. And what actually we are doing with this biotechnology and how it helped us. So that they are saying here. So this biotechnology changed our daily life as its products brought qualitative improvement in health and food production. So because of this biotechnology and all, so we are getting some qualitative. So with respect to health wise, some qualitative improvement we are observing. With respect to food production also, some qualitative improvement we are observing. So these are some important sentences from this particular paragraph, and please go through this particular unit introduction paragraph also. Now we are going with the main chapter, that is the biotechnology principles and process. And the biotechnology is also called as recombinant DNA technology. It is also called as genetic engineering, and it is also called as DNA manipulation biotechnology, because in this process genes, that means DNA, are undergoing manipulation, means some changes we are doing. That's why it is called as genetic engineering or DNA manipulation or simply recombinant DNA, because here the DNA from different sources we are combining. That's why it is called as recombinant DNA or chimeric DNA. So this is what we are actually doing in this biotechnology. But initially in the old biotechnology, so these gene manipulations were not performed, and there actually scientists tried to get some betterments in the existing varieties only. So whatever the strains of bacteria or varieties of plants or breeds of animals, whatever they are there, so within those they are trying to get some improvements because of different processes. That's it. But there technically they did not aimed at the gene manipulations and all. So that's why these all will come under the traditional or old biotechnology. So generally under this we are talking about the bread industry, and then alcohol, that wine industry, and then curd preparation. So all those things will come under the old or traditional biotechnology. But under the modern biotechnology, so all the gene manipulation related things, and even the test tube babies formation and the DNA vaccines and even the artificial synthesis of DNA or the gene, all the things will come under this modern biotechnology. Some examples also they have mentioned in NCERT, so please go through this. Under the older traditional biotechnology, we are talking about the curd, bread, wine, organic acids, all these things we are studying. But under the modern biotechnology, we are studying about this human insulin production mainly. Okay, this is where actually we have started exploiting this particular biotechnology. So actually this human insulin is the protein belonging to human, but it is produced from the bacterium called Escherichia coli. But still the same protein can be used in the human, and still there is no problem to human. So in this way we are using this one, and even test tube babies also. So they are actually formed by in vitro fertilization. These are also examples for modern biotechnology. And synthesizing a gene, that means artificial synthesis of gene and using it for various purposes, and developing DNA vaccines, and even correcting a defective gene. Correcting a defective gene means gene therapy. So all these are examples under modern biotechnology. So anyhow, there is a separate chapter called as applications of biotechnology in various fields. So there also you can study many more examples related to this particular biotechnology. Now we will focus on this, a definition of the biotechnology. So European Federation of Biotechnology, they have given a definition, and in this definition both the traditional view and also modern molecular biological view, both of them are combined. So what is the definition? So biotechnology is the integration of natural science and organisms, cells or parts thereof and molecular analogs ultimately for two things, either to get the products or to get the services. So this is what about the definition that was given by EFB with respect to the biotechnology. Clear? And this, if you are observing these principles of biotechnology, this biotechnology is actually having two core techniques. One is the genetic engineering, other one is the bioprocess engineering. What actually we are doing in the genetic engineering? In the case of genetic engineering, gene is getting engineered so that different modifications, chemical modifications we are doing to this genetic material like DNA and RNA, so that from that, so with respect to our aims and all, so we are using them in various purposes. But ultimately what we are doing is gene engineering. Genes we are changing. So at the level of the gene, we are changing the nucleotide sequences. So in this way we are doing, so that's why all these will come under genetic engineering. And in the case of bioprocess engineering, actually at the end of this particular chapter, they are talking about the bioreactor. So in the case of bioreactor, we are converting raw materials into various desired products. And there we are using some cells or microbes. Yes or no? And there what actually we are doing is, whatever the cell or microbe we want to grow, only that has to grow. If other things are growing there, that is called contamination. So by using this bioprocess engineering, we are making it contamination free. That means there is no contamination. What is the meaning of it? Whatever the cell or microbe we want to grow, that only will grow, so that there is no unwanted growth of unwanted organisms. So this is what we are maintaining. So in this way, sterile ambience we are maintaining. So contamination free conditions we are maintaining. For all these things, we need some bioprocess engineering techniques. So this is what this is also part of the biotechnology, and these are very important for the bioreactor related informations. Clear? And after that, in the NCERT, they have given a paragraph, and in this paragraph they are actually talking about differences between asexual reproduction and sexual reproduction. In the case of asexual reproduction, there is no crossing over. So because of this, what happens? The parent and the next generation individuals are exactly same. But in the case of sexual reproduction, so crossing over is taking place and many other types of recombinations are taking place. Because of this, what happens is the parental genotype and then the offspring, they are not 100% exactly same. At least a few variations we are observing. This is what actually the basic information they are saying here. And then finally, here they are talking about one important point. In the case of principles of inheritance and variation, we studied about a concept called linkage. What is the concept of linkage? If two genes, they are very closely present on the same chromosome. Suppose this is a chromosome and here two genes are present very closely. Then what happens because of the linkage? If two genes are present very closely, there is a more tendency that there is a more chance that both the genes are going together into the next generation. This is what actually we are talking in the linkage. Suppose between these two genes, one gene is a desired gene, other one is undesired gene. Now what happens is, if you are using the traditional breeding methods, can you separate these two things? No, it is highly difficult. So in the case of traditional breeding methods, along with the desired genes, undesired genes are also going to next generation. So how we can overcome this particular problem? So this can be overcome with the help of recombinant DNA technology or biotechnology. And here what we will do? Here what we do is, whatever is the desired gene is there, only desired gene we will take. Only desired gene we will isolate, and this desired gene will be introduced into the host. So because of this, what happens? There is no chance of entry of undesired gene. So in this way we can overcome those particular problems. That's why biotechnology is very useful in introducing desired genes into the target organism. This is what they are saying in this particular paragraph. Okay, and then before directly going into the content, so just behind that unit introduction, in the next page of the unit introduction, there they are talking about some scientists. Here there they are talking about actually two scientists. One is Herbert Boyer, other one is Stanley Cohen. Now let us study their contribution, because immediately after this we need to go into that content. Herbert Boyer. So what actually he did, he worked on E. coli bacterium, and from this E. coli bacterium, he isolated some restriction endonucleases. And what is the function of these restriction endonucleases? These restriction endonucleases will recognize a specific sequence, and then they are cutting that specific sequence at a particular location. So in this way, with the help of these restriction endonucleases, we can cut a DNA at a specific sequence, and later on, again using a different enzyme called ligase, again you can attach them. So in this way, he identified some enzymes which are useful for cutting the DNA and attachment of DNA. This is what the contribution of Herbert Boyer. Now we are studying about the contribution of Stanley Cohen, and what actually he did is he studied about the plasmid isolation. So what are the plasmids? Plasmids are extra chromosomal circular double stranded DNA molecules that are floating freely in the cytoplasm of certain bacteria, many bacteria. This point I told you many times. Certain bacterial cells, because all the bacteria do not have the plasmids. All the bacteria do not have the plasmid. That's why certain bacteria or many bacteria have plasmids. And these plasmids are small ring-like structures. That's why the ringlets of DNA they are saying. Understood or not? And these plasmids are autonomously replicating. That means they can divide on their own without depending on this particular nucleoid or genomic DNA. This is what the information related to plasmids. So now let us see the contribution of this scientist. What is the contribution of Stanley Cohen? He developed a method for isolation of the plasmid from bacteria. So how to isolate the plasmid from the bacterium, and this isolated plasmid, how you can introduce it into another bacterium. So these methods were developed by Stanley Cohen. So now these two scientists combinedly they performed some experiments, and that led to the development of this first recombinant DNA molecule and successful introduction and all the things. Okay, slowly we will study. But before directly going with that paragraph, in the NCERT they have given another paragraph. What is the overall information of this particular paragraph? In this paragraph, this is a very logical point. There are very logical points are there, so observe very carefully. Here what they are saying is, let us take a piece of DNA. And this piece of DNA, so we are introducing into a bacterium or a eukaryotic cell, whatever it is. So we have introduced. So once you have introduced into the cytoplasm, let us consider into the cytoplasm or the nucleoplasm in case of eukaryotic cell, you have introduced, that's it. And here let us imagine two conditions. You have introduced, after introducing into the cell, two options. First option is, the introduced DNA, let us call it as gene of interest or foreign DNA or alien DNA, anything you can say. So now this alien DNA or foreign DNA is introduced into a particular bacterial cell or eukaryotic cell. Now here we are having two options. First option, if it is not attached to nucleoid of bacterium or chromosome of eukaryote, if it is not attached to the genetic material, then what is the fate of this particular piece of DNA? Second one, if this alien DNA is attached to nucleoid or chromosome, then what is the fate of this particular alien DNA? These are the two important points you need to focus from this. So first, for the first one, what is the first one? First one, we are saying that if this alien DNA is not attached to genetic material, then what happens? Then it will be digested. So this introduced foreign DNA cannot survive on its own in the host cell. This is the first important point. So what is the second point? If it is attached to the genetic material, yes. If it is attached to the genetic material, whenever genetic material divides, then this piece of DNA also will divide. So in this way, it can exist in that organism, and in this way, it can move from one generation to next generation. So if you want to make many copies of a piece of DNA or alien DNA, then what should happen? It should compulsorily become part of the genetic material. Then only it will be sustaining there. Then it can divide and go to the next generations. This is what the overall conclusion. In this way, making multiple identical copies for a piece of DNA is simply called as cloning. This is what they are talking here. This is the molecular cloning. At the level of molecule level, we are talking about the cloning, making multiple copies of a particular molecule. That's why it is the molecular cloning. Understood or not? This is what the information related to this particular paragraph. And here also they are talking one more thing. Okay, you have introduced and it is attached to the genetic material, but that genetic material or that particular DNA molecule, whatever it is, it should compulsorily have origin of replication. Because of this, what happens? Since it is having the origin of replication, the host DNA will divide. And whenever that host DNA divides, then our gene of interest also will divide, because it is also part of that particular piece of DNA which is having origin of replication. This is what the information they have given here. And then we are talking about the procedure, general procedure of recombinant DNA technology. In the recombinant DNA technology, generally what we are doing is we are isolating a piece of gene from whatever the source. Please remember this piece of interest and then that plasmid, all these are based on our experiment. Different individuals will perform different experiments. So according to their experimental requirement, the piece of DNA or foreign DNA will be a different gene. And what is the vector that they are using, that is also of their choice. So but here we are talking plasmid as a vector, and let us take a piece of DNA from anywhere, that's it. So here what we need to do, first let us take a piece of DNA, we call it as gene of interest or alien DNA or foreign DNA. So that we are isolating from the whatever is our source, from that we are isolating. Then plasmid we need to isolate from the bacteria, okay, and some are artificial plasmids are present, so that we can purchase, and you can get it from some other labs, whatever it is. So ultimately you are getting this plasmid. Now these two are ready. And after this what happens is, now this gene of interest and the plasmid should be cut with same restriction endonuclease, so that same type of ends will be formed, sticky ends. You already studied, since it is a revision lecture, so I am imagining that you know the basic information related to biotechnology, so that they are having the same type of sticky ends, so that both of them can be attached with each other. So next, in the next step what happens, with the help of ligase enzyme we are trying to attach both of them. In this way recombinant DNA molecule or chimeric DNA molecule will be produced, and now this will be introduced into whatever is our targeted host, in that we are introducing, and this introduction is called as a transformation process. Yes or no? And once it is going inside, then we are going for the selection of recombinants. We need to select it because many combinations are possible. I will tell you under the PBR 322 selection and PUC 18 selection. I will tell you that blue white colony screening, all these are there. No, there I will tell you. So we need to screen them, because we are getting many chances out of this. So what are the recombinants that are having our gene of interest? All these things we need to identify, and once we are identifying, then we can propagate them, we can grow them for increasing number, for the increasing the number. So then finally you can collect that particular one, and that gene you can use it for different other purposes, or else you can express it so that you can get a product. That's what we are studying under the bioreactor. So these are some important points that we can relate with this recombinant DNA technology. This is the overview of this particular genetic engineering or recombinant DNA technology. And here first we are talking about the synthesis of the first artificial recombinant DNA molecule. This was done by Cohen and Boyer. These are the two scientists' contributions already we studied. And Boyer know how to cut the DNA and how to attach the DNA. And Cohen know how to isolate plasmid from the bacterium and again reintroduce into bacterium. So that's why these two scientists combinedly they performed all these things. And here please remember here, so in simple I am trying to explain this. So what actually they did is from the Salmonella typhimurium bacterium. Salmonella typhimurium bacterium causes a disease called as typhoid. Please remember this. Regarding this, we studied under human health and diseases. So please remember this point. And in this particular bacterium, there is a plasmid. I don't want to tell you the name of that plasmid. It is having a plasmid. In that plasmid, there is an antibiotic resistant gene. What is that antibiotic resistant gene? It is streptomycin resistant gene. But this name was not given here. Simply they gave antibiotic resistant gene. Antibiotic resistant gene. It is actually streptomycin resistant gene. And what scientists did is they have isolated streptomycin resistant gene from Salmonella typhimurium, and then they have combined it with a plasmid from Escherichia coli. They have combined this piece of DNA, this is the foreign DNA or gene of interest, is combined with a plasmid isolated from Escherichia coli. They have combined and they made first recombinant DNA molecule. And then again they have reintroduced this into E. coli. So in this way what happened? Now the streptomycin resistant gene is introduced into ultimately E. coli bacterium, which is closely related to this particular Salmonella typhimurium. Okay, not streptococcus, it is Salmonella typhimurium. Salmonella typhimurium is also gram negative, E. coli is also gram negative. In this way they are very closely related. In this way they have introduced. Originally this Escherichia coli is not resistant to streptomycin, but once this gene is introduced into E. coli, now it became resistant. In this way, they made the first successful recombinant DNA molecule. Recombinant DNA molecule also they made, and also it was successfully transferred into the host, and that host got this particular property also. That's why we are studying about this particular information. And in this particular process, this plasmid is actually used as a vehicle or a vector. That's why this plasmid is called as a vector. And there in NCERT they are talking about mosquito. Mosquito is carrying the infectious agent of the malaria, that is the malarial protozoan, from one individual to other individual. Yes or no? So in the same way, this plasmid is also helping carrying. Since it is helping in carrying, this plasmid is acting as a vehicle. That's what they are saying. That vehicle is nothing but the vector. That's what we are saying. This is what the overall overview of this particular one. And in this process, for the isolation of these things, they have used some enzymes like restriction endonucleases, which are also called as molecular scissors. And later on, for the attachment, they have used an enzyme called as ligase. And then they have introduced that into the E. coli. So all these basic techniques they have used. This is what about this particular experiment. And finally, they are saying that there are three basic steps when we are trying to modify a particular organism. What are these three particular basic steps? First we need to identify the DNA with the desirable genes. That means this is the gene of interest that you need to select. And once you are selecting this, then you need to introduce into the host. And after introducing into the host, in the host it should be maintained so that it should be transferred to the next generations. These are the important things that we need to keep in mind when we are doing this particular genetic engineering. And then we are talking about what are the various important tools that are required for this recombinant DNA technology. Please remember here what I am doing is, see here total three important tools are there. One is enzymes, next one is cloning vector, next one is competent host. In the NCERT they have given randomly. But what I will do is, from the NCERT, whatever the information that is related to enzymes is there, entire information we will cover at once. And whatever the vectors that they have mentioned in NCERT, all the vectors we will cover next. And finally, we will talk about the competent host. So in this way, when we are going with this, one by one we are going, and when we are going through this particular concept of tools, we are actually covering different individual points given in the NCERT. So in this way, I am not going through NCERT sequence exactly, but I am trying to cover the entire NCERT information. So under the tools, first we are talking about the enzymes. What are the various enzymes that are required? Lysing enzymes, cleaving enzymes, DNA ligases, DNA synthesizing enzymes, and many other enzymes are there. But we are confining ourselves to NCERT content only. Okay, other enzymes are not needed. Simply we will see the NCERT information. First one are lysing enzymes. Where actually we are using lysing enzymes? If you want to isolate a DNA from a cell, first you need to break the cell. In the case of bacteria, cell wall is present. To lyse the bacterial cell wall, lysozyme enzyme is required. If you want to isolate DNA from the plant cell is having the cellulose. So that's why you need a cellulase enzyme to break plant cell wall. If you are isolating DNA from the fungal cell, then what you need to do? You need to use the chitinase enzyme so that that fungal cell wall will be broken. So in this way, for the lysis of this particular cell wall of corresponding bacterial cells, different enzymes are used. So these are also enzymes that are used in recombinant DNA technology. But remember this one, these names once again we will study under isolation of DNA. Okay, next one. They are of one type of enzymes. Other type of enzymes are cleaving enzymes. What these enzymes will do? They will break the phosphodiester bonds. So these enzymes are called as exonucleases and endonucleases. Why they are called as exonucleases? They are acting on the last phosphodiester bonds. If the enzyme is acting on the last phosphodiester bond towards 5' end, that is called as 5' exonuclease. If the enzyme is acting on last phosphodiester bond present towards 3' end, it is called as 3' exonuclease. So these are also some enzymes. Next one, endonucleases. These will not target the last phosphodiester bonds. Except these two last phosphodiester bonds, any of these internal phosphodiester bond can be broken. So that's why these are called as endonucleases. Inside they are breaking this. In these endonucleases also, there is a separate group that is called as restriction endonucleases. And what these restriction endonucleases will do? They are recognizing a specific sequence, and that sequence only they will cut. If the recognition sequence is not there, they will not cut the DNA. So in this way, they are very much specific in their action. And these enzymes are also called as biological scissors, and they are also called as chemical scalpel because they are exactly cutting at some particular locations. This is what they are saying. And now the point is, in the restriction endonucleases, from where exactly we are getting the term called a restriction? You need to understand this. Some students are thinking that restriction means they are confining only to one sequence. That's why it is restriction. No, it is not like this. Actually, the meaning of restriction means what? Stopping. So restriction enzymes, what actually they are stopping. So you need to understand the concept. I will come to this paragraph later. First I will explain you the important information, observe carefully. And this is called as a restriction modification system. What is restriction modification? For restriction there is an enzyme, for modification there is an enzyme. What these two enzymes will do? Observe carefully. Suppose this is the bacterium, and already you know about some viruses called as bacteriophages. What are bacteriophages? These are the viruses that infect the bacteria. So now we are talking about the bacteriophages. Bacteriophage is infecting this bacteria. Once it is infecting the bacteria, what it will do? The bacteriophage will introduce its DNA into the bacterium. So in this way it has introduced its DNA into bacterium. Okay. And once it is introducing its DNA into bacterium, what happens? That viral DNA will become part of bacterial DNA and it will start synthesizing new viruses. This is what the general procedure. But here there are some bacteria, and these bacteria they are preventing the viral growth, viral propagation. How they are preventing, how they are stopping, how they are restricting. Stopping means restricting. So that we will see here. So virus is attaching to bacteria and viral DNA is released into the bacteria. Once viral DNA is released, now what happens? There is a specific enzyme here, and that enzyme will come and cut the viral DNA. And what is the enzyme that is cutting viral DNA? That is the restriction endonuclease. How it is able to cut here? So within this viral DNA, there are some sequences, and these sequences are recognized by restriction endonucleases. And these restriction endonucleases will cut at that location so that viral DNA is broken into fragments. Once viral DNA is broken into fragments, now it cannot combine with bacterial DNA. So in this way, bacterium is able to stop viral growth. That's why this restriction endonucleases are restricting the viral growth. So this is what the enzyme is. So this is what one important enzyme is. I hope now you understood why this is called restriction endonuclease. It is acting as endonuclease and it is restricting the viral growth. That's why it is the restriction endonuclease. That is one enzyme. What is the other enzyme? So now you understood about restriction, but what is modification? Now understand the question, then only you will understand the answer. See, this restriction endonuclease is present inside the bacterium. And what is its function? Recognize a specific sequence and cut it. Whether it is viral DNA, bacterial DNA, it doesn't know. Please understand this, it doesn't know. If the recognition site is there, it will go and bind there and it will cut there. This is what the function of restriction endonuclease. So now here for the bacterium there is a problem. What is the problem? It is producing restriction enzyme, okay, but bacterial DNA is also having some restriction sites for that particular enzyme. Now the problem is how bacterium will protect its own DNA from the breakdown by that particular restriction endonuclease. This is the important point. So for this, there is a separate enzyme called as a modification enzyme. So there is an enzyme. What that enzyme will do? See here, there are some sites here, and these sites are recognized by the enzyme, and enzyme can cut here. But bacteria should protect its own DNA. If bacterial own DNA is broken, then what is the use of doing all these things? So how bacterium is protecting its own DNA? By modifying these particular bases. The sequence is same only, but some nucleotides are modified by attachment of some chemical groups, mainly by methyl groups. So methyl groups are added at the restriction site. Once methyl groups are added at restriction site, enzyme cannot recognize that site. So in this way, bacterium is protecting its own DNA. But this methylation process is not happening for viral DNA. So that's why enzyme is immediately cutting the viral DNA. So in this way, in the restriction modification system, there are two enzymes. One enzyme is modifying bacterial DNA, not viral DNA, bacterial DNA. In this way, it is protecting bacterial DNA. And other enzyme is restricting the viral growth by cutting the viral DNA. So that's why it is the viral. So that's why it is doing the viral restriction, viral growth restriction, viral growth stop. That's why it is the restriction endonuclease. So this is what they are talking here. So here in the NCERT, they are saying that two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. Two enzymes. They are restricting, stopping the growth of virus in the E. coli bacteria. One enzyme is adding methyl group to DNA modification. By the addition of the methyl group to DNA, whose DNA? Bacterial DNA, not viral DNA, bacterial DNA. It is protecting bacterial DNA. While the other is cutting the DNA, whose DNA? Viral DNA. So here this is adding methyl group to bacterial DNA, while it is cutting the viral DNA. So these points also you need to understand. In both the locations it is DNA, but whose DNA if you don't know, then there is no use. So observe this carefully. And later these are called as restriction endonuclease. Actually this one cutting the DNA is called as restriction endonuclease, and this is the restriction modification system. Though it was not clearly explained, since these sentences were given, you need to understand this information. Clear? And how exactly these enzymes are working here. So all these enzymes, all the restriction endonucleases will recognize a particular palindromic DNA sequence. In general here, for the palindromic term, here we are talking about 2 points. Many students are not observing carefully. So I will try to explain this, understand carefully. With respect to palindrome, we are talking first one, palindromic words. Second one, palindromic DNA sequence. There is a small difference between these two. You need to understand the difference. What are palindromic words? So these are Malayalam, Madam, Noon, Rota, Radar, all these words if you see. For the same word, left to right you can read or right to left you can read. You will get exactly same spelling. That means sequence is exactly same on both the sides. From left to right and right to left exactly it is same. So these type of words are called as palindromic words. That is correct. Okay. But our aim is palindromic DNA sequence. When you are doing palindromic DNA sequence, there is a small difference here. There is a small difference here. So observe it very carefully. So here what you are doing, in one strand 5' to 3', suppose GAATTC is there. Are you reading the same strand sequence in opposite direction? No. You need to read the sequence of opposite strand from 5' to 3'. This is the difference. Here the same word you are reading in left to right, right to left. But here it is not the same sequence in the single strand. Here in one strand if you are reading from 5' to 3', for the same segment in the opposite strand you need to read from 5' to 3'. Then you will get exactly same sequence. Or in one strand you can read from 3' to 5'. In the other strand for the same segment read from 3' to 5'. You will get exactly the same sequence from 5' to 3' or 3' to 5', whatever it is. Fix it for one strand. In the other strand also you need to follow the same direction. Then you will get exactly the same sequence for that segment. And if such sequences are present, they are called as palindromic DNA sequences. And restriction endonuclease will recognize such type of palindromic DNA sequences. That is what they are saying. So this is what. And in this sequence, some may be cutting exactly at the center, some may be cutting slightly away from the center. Slightly away does not mean that 100 nucleotides away. No. Within this site, somewhere here, somewhere here, somewhere here. But the point is, if it is cutting between G and A of one strand, here also it is cutting between G and A only. So in this way they are cutting. So in this way they are actually acting. And here we are talking about eco R1 restriction endonuclease recognize it, and it is actually recognizing 5' GAATTC 3'. And now here it is exactly cutting between G and A. Understood or not? So in this way it is producing. So in this structure if you see, for this one it is 3' means this side it is 5', and these 4 nucleotides are single strands and they are actually hanging here. That's why they are called as overhangs. And which end is hanging? 5' end is hanging. That's why it is the 5' overhangs. And here these type of nucleotides, this AATT and this TTAA are complementary to each other. Again they can attach with each other, and that's why they are called as sticky ends or staggered ends. So this is what some information we are studying. Anyhow, regarding sticky ends, once again I will tell you. And here, and after completion of this basic information, now we will see the NCERT content. Here they are talking about some first restriction endonuclease is HIND2, and this Roman number 2 indicates it is a type 2 restriction endonuclease. There are type 1, type 2, type 3, type 4 different restriction endonucleases are there. And please remember, in our NCERT content they are not talking anything about these types. And please remember, in general in the recombinant DNA technology we are using type 2 only. Generally we are using type 2 only, but very rarely other types can be used, but in our syllabus we are using only type 2. So that's why every information we are studying is with respect to type 2 only. Clear? And here it is a HIND2 is the first restriction endonuclease that was isolated. From whom? From which organism? H means what? Haemophilus. IN means what? Influenzae. So first restriction endonuclease was isolated from which organism? Like this also they can ask. It is from Haemophilus influenzae. Anyhow, under the nomenclature I will tell you. So please remember this. And here, this is what. and this particular enzyme is recognizing a particular 6 base pair sequence. It is recognizing a 6 base pair sequence, and in this 6 base pair sequence also it is cutting exactly at the center. Since it is cutting exactly at the center, there are no single nucleotides, means unpaired bases. So this type of ends are called as blunt ends. These are called as blunt ends. So this is some information that we are talking here. Nowadays scientists have isolated so many more than 900 restriction enzymes. They have isolated from over 230 strains of various bacteria. Okay, this is what some basic information that they are talking with respect to this one. And then we are talking about the nomenclature of the restriction endonuclease. In the NCERT they are talking the nomenclature with respect to EcoR1. So quickly I will tell you. In this nomenclature, first letter E, capital letter, and this E is obtained from the first letter of the genus name. That is the first letter of this Escherichia, E. And the next two letters CO are actually the first two letters CO. First two letters of this species name. So in this way, from the coli, first two letters are taken here, and this R was isolated from the strain name, that is RY13. From that R was taken here, and this Roman number indicates the sequence in which these restriction enzymes were isolated from exactly the same strain of bacteria. This is what. So in this way, they have given this information in the NCERT. But observe for Hind 3, Hind 2, whatever it is, this Hind 2 here we have studied, right? And here I have writing about the Hind 3, whatever it is isolated from H means what? Haemophilus. IN means what? Influenzae. RD, that D is from RD. In this way, first enzyme will be 1 and second will be 2, but please remember here directly the name was given 2. So it is 2, and the next will be the 3, next will be 4, next will be 5. Likewise, the sequence in which these were isolated from that particular strain. And here if you see the solvent, it was isolated from Streptomyces albus. But here we are not writing strain name, directly the Roman numbers 1, 2, 3 we are giving. Okay. Next one, if you see the PVU2, here it is isolated from Proteus vulgaris, PVU. And here also strain name is not there, and directly we are writing this particular Roman number. Clear? But anyhow, in the NCERT they have given the explanation with respect to EcoR1. At least that one you please remember. Clear? Next one. Next one is regarding sticky and blunt ends. If the enzyme, restriction enzyme is cutting exactly at the center of this particular recognition site, then we will get blunt ends. If it is cutting slightly away, maybe on this side or that way, anywhere, so then we will get this type of ends. And in these ends, what happens is there are some unpaired nucleotides, and these two can again attach with each other. So that's why these type of ends are called as sticky ends. So EcoR1 is producing sticky ends, but Hind2 is producing blunt ends. At least these informations are related to NCERT, so please remember this information. And with respect to EcoR1, the recognition site is 5' GAA-TTC, and here it is cutting between G and A. And in the overhang, how many nucleotides are present? Each overhang is having 4 unpaired nucleotides. In these unpaired nucleotides here, 2 purines, 2 pyrimidines are present like this. Is it clear? And overall, if you see in the recognition side, 6 base pairs are present. That means 12 nucleotides are present. Out of this, anyhow in each pair, one will be purine, other will be pyrimidine. So definitely 6 will be purines, 6 will be pyrimidines. And how many hydrogen bonds are present? Observe carefully here. Between G and C, 3 hydrogen bonds, A and T, 2 hydrogen bonds, A and T, 2 hydrogen, A and T, 2 hydrogen, A and T and A, 2 hydrogen, C and G, 3 hydrogen bonds are present. So that's why 3 plus 3, 6, and 4 into 2, 8. 8 plus 6, it will be 14 hydrogen bonds are present in this particular recognition site. But please remember, when the enzyme is actually cutting this, it is not focusing on the hydrogen bond breakage. It is actually breaking the phosphodiester bonds breakage. So it is cutting between A and G, 1 phosphodiester bond. Here also between G and A, 1 hydrogen bond. Sorry, 1 phosphodiester bond. So these 2 phosphodiester bonds are broken. Once these 2 bonds are broken, so hydrogen bonds will be broken automatically, because they are very weak bonds. So that can be separated very easily. Enzyme is actually breaking the phosphodiester bonds. Enzyme aim is not to break the hydrogen bonds, but they also will be broken by default. So these are some important points that you need to remember with respect to this one. What are these 3' and 5' overhangs? So after the breakage, so which end is hanging here? 5' end is hanging here. That's why these are called 5' overhangs. This is a different enzyme. You don't need to remember the sequence. But in this spot, so which end will be hanging here? 3' end will be hanging. That's why it will be the 3' overhang. So which end is actually hanging? So based on that also they can ask. But with respect to eco-R1, you need to remember. So eco-R1 produces what type of overhangs? 3' or 5'? It is producing 5' overhang. So please remember this particular information. Clear? And here the action in the NCERT they have given a diagram. So here you are taking the vector DNA. On the other side, you are taking gene of interest or foreign DNA, and we are cutting both of them with the same restriction endonuclease. In this case, let us take eco-R1. So it is cutting between G and A, between G and A. So it is making this sticky end. And here also between G and A is okay, but here they gave wrong. Between A and A they have mentioned it is wrong. So cut this arrow mark, and this arrow mark you need to keep between G and A. And this arrow mark by mistake they have given, so remove that. Please remember between G and A you need to keep arrow mark so that you are getting this type of sticky end. Understood or not? So later on what we are doing? So we are connecting both of this. In this way, vector DNA and gene of interest are combined. So in this way we are producing recombinant DNA molecule. And for this attachment, so we are using the ligase enzyme. It is also called as molecular glue, so that it is helping in attachment of these sticky ends. Clear? So this is how recombinant DNA is produced. Same another diagram. Foreign DNA on one side, vector on the other side. Both of them are cut with the same restriction endonuclease. So that here, let us consider that this is our piece of DNA that is required for cloning, and this is the vector that got opened up. This plasmid is initially a circular molecule. Because of the breakdown, it became the linear. And now this piece is introduced here, and now with the help of ligase it is attached. And in this way, recombinant DNA or chimeric DNA molecule is produced. Now it is transformed into the bacterium. Yes or no? We are transforming this into the bacterium, and then now we are allowing for the division. So when it is dividing, what happens? Along with the plasmid, our piece of DNA is also dividing. So in this way, making multiple identical copies is called as a cloning. So this is the cloning. And this cloning we are doing inside the organism. So that's why it is in vivo cloning. In vivo cloning. Suppose this piece of DNA, you are making multiple copies in the PCR reaction, that is in vitro cloning. You can do the cloning in any way, but this is cheaper. So if you are introducing into the bacterium and growing, this is cost effective, it is a cheaper. But if you are doing this in the PCR, that is costlier. That is the difference, that's it. Clear? So in any way we can do this one. This is some information regarding the enzymes, and in the enzymes we are talking about further things. And here we are talking about DNA ligase. It is also called as a molecular glue because it is able to attach sticky ends. Since it is able to attach the sticky ends, we are calling this one as molecular glue. This is what, in this way we can attach these molecules. And then DNA synthesizing enzymes regarding the Taq DNA polymerase, that is also we are using in the recombinant DNA technology. So these DNA synthesizing enzymes are useful for the synthesis of DNA. So DNA polymerase, what it will do? It will utilize DNA as template and directly it will synthesize the DNA. But in some cases from the RNA we need to produce the DNA. In that case we are using reverse transcriptase enzyme. Yes or no? So all these enzymes are useful ultimately for the synthesis of DNA molecule. With that, under the tools, we studied total 3 enzymes, vectors, and competent host. Enzymes we have completed. Now under the tools, we are studying the second one, that is the vectors. So in the NCERT, these vectors are given at different locations, and all this information we are studying at once. Please observe carefully. And vectors, what are these vectors? These are called as vehicles or carriers. Carrying what? Carrying gene of interest from one location to other location. That other location is nothing but the host. So now these vectors or vehicles are useful for transfer of gene of interest into the host. This is what their function is. And now there are different types of vectors mentioned in the NCERT. Plasmids they have mentioned, bacteriophage vectors they have mentioned, artificial chromosomes also they have mentioned in different chapters. I am combining all this information. First we will focus on the plasmids. And here under the cloning vector, they are talking about bacteriophage vectors and plasmid vectors. First we will focus on the plasmid vectors. What is the copy number of the plasmid? And after completion of all these plasmids, we will go with bacteriophage vectors and what is the copy number. Only that much they give. Only that much information we will study. And now we will directly go with this particular plasmid vector. So what are the plasmids? Plasmids are extra chromosomal, circular, double-stranded DNA molecules floating freely in the cytoplasm of certain or many bacterial cells, but not all bacterial cells. Very, very important. Repeatedly I am talking about this. And these are the ringlets of DNA. These are autonomously replicating DNA molecules. Autonomously means they can divide on their own without depending on this particular nucleoid. That is what the meaning is. These are the plasmids. Now we will see about the what is copy number of plasmid. What is copy number of plasmid? For a specific plasmid in a single bacterium, how many copies are present? That is called as copy number. So for this one, total how many copies are there? Total 3 copies are present. That's why it is here in this diagram. In this diagram, for this particular plasmid, what is the copy number? It is the 3. In this way, different plasmids are having different copy numbers. That's what we are talking. Let us talk with respect to PBr322 because they have given in NCERT. So this PBr322, generally it is having less than 20 copies in the cell. That means in the range of 15 to 20 it is having. So what is the copy number? If 15 are present in a cell, the copy number will be 15. That's what the copy number means. In this way different. So this is actually, since it is having less than 20, it is coming under the less copy number plasmid. But there are some plasmids whose number is actually in the range of 20 to 100, and this is the medium copy number. But there are some plasmids whose copy number is more than 100, so they will come under high copy number plasmid. One most important example is PUC18, PUC19. Why we are talking about PUC18, PUC19? Did they mention in NCERT? No, they have not mentioned this name. Even though name is not mentioned, blue white colony screening they have mentioned in NCERT. That is with respect to these only. So you should know about these things. So in this way, this is what about the copy number for the plasmid. And now directly in the NCERT they have given one important example, that is PBr322. In the PBr322, P indicates plasmid, B indicates Bolivar, it is name of scientist, R indicates Rodriguez, it is also name of scientist. So this PBr322 name was given based on names of the scientists. Then what is this 322 indicates? This 322 indicates the serial number. Actually they have developed different plasmids. So in this way, by giving the number to these plasmids, we can differentiate one plasmid from the other plasmid. All these are PBr only, but they are having different names because actually in the DNA structure of these particular molecules, the nucleotide sequence is slightly different. So distinguish, how we can distinguish these plasmids from one another? By giving the various numbers. So in this way, 322 is a number that was given, is a number, it is just a number that was given to distinguish this from the other plasmids that were developed in the same laboratory. Is it clear? So this is what some information is. Actually in this particular plasmid total 4361 base pairs are present, but this information you don't need to remember. Clear? But this is the PBr322 diagram that was given in the NCERT, and the most common mistake done by the students is how many restriction enzyme sites are present? 1, 2, 3, 4, 5, 6, 7. Students are saying that so PBr322 he is having 7 restriction endonuclease sites. This is the blunder mistake done by not just 7, this one is also there 8. They are saying 8. This is a blunder mistake done by majority of the teachers also. Here in the NCERT they have given very few restriction enzyme sites, but if you see original PBr322 complete plasmid map, you will find more than 50 restriction enzyme sites. You will find more than 50 restriction enzyme sites, and here they have not given that much. We should be thankful to NCERT. They have not given those many, because if they are giving those many, we need to remember all the things. So that's why, to avoid the confusion and then to avoid the burden, in the NCERT they have given very few restriction endonuclease sites. That's it. Suppose you are anywhere you are encountering a question that how many restriction endonuclease sites are present in this PBr322, the answer will be more than 50 are there. More than 50 are there. But if they are giving answer as 8 based on this diagram, that will be wrong. That will be 100% wrong. Here they have mentioned only few. This point very clearly I am telling you, but still students are having the doubt. So please observe this carefully. They have not given the complete map here. Only few restriction enzyme sites they have given. That much is enough here. Clear? This is what. So in this particular PBr322 vector map, what are the important points we need to remember? First one is it is having ORA. This ORA is origin of replication. This is the place from where replication of DNA begins. This is one important thing. Second one, it is having ROP. What is this ROP? Replication of plasmid. So ROP is the gene, is having some genes that are producing some proteins, and these proteins are actually needed for replication of the plasmid. That's why it was named as ROP. and along with this it is having two selectable markers. One is ampicillin resistant gene, other one is tetracycline resistant gene. And please remember what this ampicillin resistant gene will do. This ampicillin resistant gene will produce a protein, and that protein will interfere with the action of ampicillin, so that if this particular gene is present in the bacterium, ampicillin cannot kill the bacterium. In this way, this gene and product is giving resistance to ampicillin. Understood or not? Same way here, tetracycline resistant gene produces a protein. That protein will interfere with action of tetracycline, so that tetracycline cannot kill the bacterium. These are the functions of these genes. But many students are making mistake here. What many students are thinking? Ampicillin resistant gene produces ampicillin. That is a blunder mistake. Tetracycline resistant gene produces tetracycline. That is a blunder mistake. Don't do this type of mistakes. Try to understand the concept. These are the antibiotic resistance genes. These genes are producing proteins, and those proteins are interfering with the corresponding antibiotics, so that these antibiotics cannot do anything to that particular bacterium having the corresponding genes. These are the selectable markers. So such type of antibiotic resistant genes and some other genes can be used as selectable markers during the selection of recombinants in the recombinant DNA technology. That's why we are calling these as selectable markers. Understood or not? Next point is, see here. This is called as, in general it is called as MCS, multiple cloning site, but in NCERT they have not given. It is called as multiple cloning site, and here also there are some restriction enzyme sites, and here also you can introduce your gene of interest. But what is suggested? It is suggested to introduce gene of interest anywhere within the selectable markers. Within the selectable markers, if you are introducing the gene of interest, then it will be easy to select. That's why they are called selectable markers. If you are not introducing in selectable marker, if you are introducing somewhere in selectable marker, then it will become difficult for you to select which one is recombinant, which one is non-recombinant. So that's why it is suggested to introduce gene of interest. So anywhere, BAMH1, SOL1, or PV1, PST1, as per NCERT I am saying, because these are selectable markers. Introduce anywhere here. Suppose you are introducing at these three locations are here, again it will create a problem. So this is what the significance of these particular locations. So please observe each and every point related to this very clearly. Okay. Next one, these are the points that we need to observe carefully. With that, PBR 322 vector information is completed. Selection based on PBR 322, I will tell you separately. Here we are talking only the vectors. Vector information is completed. Next one is PUC 19 or PUC 18 vector. So anything you can use it here, but this name was not mentioned in NCERT. But you should remember, because blue and white colony screening is based on this one only. That is not based on PBR 322. Blue and white colony screening is based on PUC 18 or PUC 19, and here what is important, here also it is having origin of replication. Okay, but here selectable markers are, one is ampicillin resistant gene, even though they gave AMPA at the top, you need to write R. This is ampicillin resistant gene is one selectable marker. Other one is LAC-Z, and multiple cloning site, MCS, multiple cloning site is present within the LAC-Z. So gene of interest we are introducing within the LAC-Z. Gene of interest we are introducing in the LAC-Z. So please remember, for the PBR 322, what are the selectable markers? Ampicillin resistant gene and tetracycline resistant gene. But for PUC 18, PUC 19, what are selectable markers? Ampicillin resistant gene and LAC-Z. It is not tetracycline resistant gene. These 2 points you need to remember. Based on this only, selection, whatever they have given in NCERT, we can easily understand. Okay. With this, regarding the plasmids, it is completed. Under the artificial chromosomes, bacterial artificial chromosome, yeast artificial chromosomes, I am not going into the details. These names actually we studied under molecular basis of inheritance. There, under the human genome project, there we studied that. So in the sequence annotation method, so scientists have isolated the chromosomes, and then they have broken them into small fragments, and each fragment is attached to this BAC-YAC. Based on their sizes, they were attached to BAC-YAC, and these are acting as vectors, and these are introduced into corresponding host. BAC is introduced into bacteria, YAC is introduced into the yeast. In this way, they are propagated there. So these are also used as vectors. So please remember this information. Okay. Next one, vectors for cloning the genes in plants and animals. This point was given separately. So please observe carefully. First we will see what are the various vectors that are used for gene cloning in the case of plants. Here we can use bacterial plasmid vectors and we can use plant viruses as vectors. Plant viruses also can be used as vectors. First we will see bacterial plasmid vectors. There we are talking about the TI plasmid. What is TI? Tumor inducing plasmid. This is also a plasmid, and this is present within the Agrobacterium tumefaciens bacterium, and this bacterium is called as natural genetic engineer, because whatever the genetic engineering techniques we are using, exactly same thing is actually used by this particular bacterium. See, many of the techniques we are actually learning from these natural organisms only. Okay, and please remember, this particular bacterium is causing a disease called as crown gall disease. This is called like a tumor. These are like tumors within the plant. This is called as a crown gall disease, and this plasmid is called as tumor inducing plasmid. And here in the NCERT properties they have not given. In simple I will try to tell you, so observe carefully. In this particular TI plasmid there is a tDNA region. There is a tDNA region. What are present in tDNA region? Left border is there, right border is there, and between this there are some genes for oxygen synthesis, there are some genes for cytokinin gene synthesis, and there are some genes for wapine synthesis. Please remember, these wapines and genes will produce the wapines, and these wapines are used by the Agrobacterium tumefaciens. Okay, next one. These oxygens and cytokinins, they will cause the they will accelerate the cell cycle process, and they will cause the cell division. So that what happens, wherever this infection is taking place, there cell division will occur. There is a tumor formation. So these are needed for tumor formation. So because of this tumor formation, more cells are present, and in these cells more wapines are produced, and this bacterium is happily growing. This is what naturally happening. This is what naturally happening. But please remember, when we are using this TI plasmid as a vector, then you should not use these particular things, because they are causing damage to the crop, damage to the plant. So that's why what we are doing is, we will take the TI plasmid, we will take the tDNA region, but we will remove these disease causing genes. We will remove the disease causing genes, and in place of that we will introduce our gene of interest. We will introduce our gene of interest, and in this way we are preparing this TI plasmid recombinant DNA, and this TI plasmid will be introduced into Agrobacterium tumefaciens, and now this will be given infection to the targeted plant. Generally it is infecting the dicot plants like tobacco and many other things, right? Anything. So in this way it is infecting the dicot plant, so we are giving infection. Then what happens? Now this tDNA portion will be introduced into targeted cell. So, but please remember, in this tDNA, these disease causing genes are not there, gene of interest is there. So now this tDNA portion will become part of targeted DNA. So in this way, in this TI plasmid, within this tDNA portion, what actually we are doing? We are introducing our gene of interest. We are removing the disease causing genes, and then we are infecting the plant. Now it will become the part of only tDNA. Please remember, entire TI plasmid is not introduced. Only tDNA portion is attached to this particular plant chromosome. So that now, since our gene of interest is going into plant chromosome, now it will start expressing there. And if it is the original case, if you are talking, this tDNA is introduced there, and it is causing the tumor there, and it is called crown gall. Yes or no? But when we are using it for the recombinant DNA technology, disease causing genes we are removing. That's why it will become non-pathogenic or disarmed pathogen. It is a pathogen originally, but it is disarmed. Arms means what? So let us consider this. These are the arms, right? So disarmed means hands are removed. That means now it cannot cause the disease. So in this way, disarmed pathogen. Originally it is a pathogen, but now it became disarmed. So disarmed pathogen is used as vector. So these are some important examples. In the same way, RI plasmid, Agrobacterium rhizogenes, is having root inducing plasmid, and here it is causing the hairy root disease. So many roots are produced, and here even this plasmid also can be used as a vector, and here also disease causing genes are removed and gene of interest is introduced in that location. In this way, that is also used. These are plasmid based, and now even plant viruses can be used as vectors. But please remember, viruses will cause the disease, so as it is the original virus, you cannot use as a vector. So in that virus, some genes are there which are helping in transfer of genes from the virus to plant. Those genes we are keeping, but disease causing genes we are removing. Since we are removing the disease causing genes, now again these are coming under disarmed pathogen vectors. So in this way, plasmids can be used as vectors, even the viruses can be used as vectors, but please remember, we should make them disarmed. Then only we are using these disarmed pathogen vectors for this particular process. Next one. for the vectors that are used for genes cloning in the case of animals. In the case of animals also we can use some viruses. So but here also we need to again make them into disarmed pathogen vectors. Then only we can use them for transfer of this gene of interest into the targeted animal. So in this way we are using different types of different types of vectors. So bacteriophage vectors are also there. What is copy number for bacteriophage vector? So bacteriophages can be used as vectors. And here what is the copy number? A virus called bacteriophage is infecting the bacterium. Once it is infecting the bacterium, inside the single bacterium it is producing thousands of viruses. So inside a single bacterium, how many viruses are produced? That number is called copy number for the bacteriophage. So generally if you see, when compared with the plasmid, these bacteriophages will have more copy number. So that's why in some cases, if you want to get more copies of DNA in a very less time, so even these bacteriophage vectors are also used. This is what the general basic information. So here different types of bacteriophage vectors like bacteriophage lambda, many are there, but not given, any of these names were given, not given in NCERT. So we are not going into those details. Clear? Next, anyhow, regarding the artificial chromosomes, also I told you BAC, YAC. So these are the various vectors that we are studying under this particular heading of tools. Under the tools, under the heading of cloning vectors, we are studying this. So under the heading of tools, till now we have completed the enzymes and the cloning vectors. And now we are pending with this competent host. Now we will study about the competent host. What is this competent host? Please remember, in the NCERT, we are talking about the PBR 322 vector based selection and PUC 18 or PUC 19 based blue white colony screening. In both these cases, for both of these vectors, the host is the E. coli. So we are talking only about the E. coli. So here in this E. coli, what happens is, E. coli is a bacterium. This bacterium is having outside cell wall and the inside plasma membrane is there. And please remember here, in the process what actually we will do is, we will take the bacterium and we will take this recombinant DNA and we will combine both of them in a single tube. Now please remember, this recombinant DNA has to pass through bacterial cell wall and then it has to pass through the plasma membrane. Then only it will enter into the cytoplasm of the E. coli, yes or no? So now here very clearly they have mentioned in the NCERT, observe the content. How it will pass through the cell wall first? How it will pass through the cell wall first? Please observe carefully. This E. coli bacterial cell wall is negatively charged and DNA molecule is also negatively charged. Yes or no? DNA molecule is also negatively charged. So negative repulsion will occur. So this DNA cannot easily pass through the bacterial cell wall. So that's why this bacterium is treated with divalent cation like calcium. So that's why generally we are using the 0.1 molar calcium chloride. Generally we are using 0.1 molar calcium chloride we are using. So here what is the aim? This calcium will bind with cell wall of bacterium. Cell wall is negatively charged, calcium is binding so that it is neutralized. So now in the cell wall, again already in the cell chapter I told you it is a porous structure, it is having pores. So once it got neutralized, now this negatively charged DNA, recombinant DNA can easily pass through these pores. In this way it is passing through the cell wall. So that's why we are doing this divalent cation treatment, so that this negatively charged DNA molecule, which is a hydrophilic molecule, can easily pass through the cell wall. So once it is crossing the cell wall, between cell wall and plasma membrane there is a periplasmic space. Up to this it is coming. And now there is a second barrier. What is that? Plasma membrane. So this DNA being negatively charged molecule, being a charged molecule or negatively charged molecule or hydrophilic molecule, it cannot easily pass through the plasma membrane. And to pass through this plasma membrane, now here we are giving the heat shock. So generally, initially we are keeping this mixture at the 0 degree centigrade. Means at the on the ice at a 4 degree technically 4. Even though you are saying ice, you are not saying 0, it is 4 degree. From the 4 degree, we are taking the tube out and keeping it 42 degrees centigrade. Because of the sudden increase in the temperature, now what happens? There is some disturbance in the plasma membrane. So because of the disturbance of the plasma membrane, there are some temporary pores formed. So through the pores from the periplasmic space, this DNA is entering into cytoplasm. So that's why heat shock is very important. So please observe carefully. This calcium ion treatment we are giving so that DNA can easily pass through cell wall. And the heat shock we are giving so that DNA can easily pass through plasma membrane, so that finally it may enter into cytoplasm. Even though you are doing this much, the chance of entry of this recombinant DNA into the bacterium is very less. Even after all these things, even after chemical treatment and heat shock also. This is what one important point that we need to understand. In this way, we are preparing the bacterium getting ready to take the external DNA. So that's why the bacterium which is ready to take external DNA is called a competent bacterium. So competent bacterium is prepared with the help of calcium chloride treatment. This is what the competent host concept is. But please remember this host, whatever we are using, it should not naturally have the antibiotic resistant genes whatever they are present in the plasmid as selectable marker. Please remember, whatever the plasmid you are introducing, that plasmid is having some selectable marker. Let us talk for PBR. Let us talk with respect to PBR322. PBR322 has ampicillin resistant gene and tetracycline resistant gene. These two genes should not be naturally present in E. coli host. Whatever the E. coli host that you are using, in that host, these two should not be present. Other genes can be present. So this is what very important. That's why here they are saying that normal E. coli cells do not carry resistance against any of these antibiotics. What is the meaning of this? Whatever the selectable markers that are present within the plasmid, exactly those selectable markers should not be present within the host. This is the compulsory requirement. If host is also having that particular selectable markers on its own, then again during the selection it will be a problem. So that's why you need to understand the significance of this particular sentence also. Anyhow, each and every sentence with words also very clearly I have explained in the regular classes, but since it is a revision lecture, so I am talking on the important points. That's it. And with this under the tools we studied about the enzymes, vectors, and also about the competent host. With this, all the three tools we have completed. Now we are talking about the transformation process. In the transformation process, what we do is, initially we are taking the mixture of this particular competent E. coli cells, and then we are adding this particular recombinant DNA molecule in one tube. And after adding in one tube, initially we are keeping it at 4 degrees centigrade. Then suddenly we are transferring to 42 degrees centigrade. At this 42 degrees centigrade, generally we are keeping for 30 seconds to 45 seconds. It is seconds, not minutes. Just for few seconds we are keeping. And again we are bringing it back to 4 degrees centigrade. Because of these temperature changes, temporary pores are formed in the plasma membrane so that this recombinant DNA can enter into the cytoplasm. That is what the aim of this particular process. And the process is called as a transformation. In simple, transformation means the taking of external DNA into the bacterial cell is simply called as transformation. Clear? This is what they are saying here. In next one, separation and isolation of DNA fragments by agarose gel electrophoresis. Gel electrophoresis. In the gel electrophoresis, gel will act as a medium. In our condition, we are preparing that gel with the help of agarose. That's why it is called as agarose gel electrophoresis. And this agarose is a polysaccharide. It is a powder that is obtained from, it is a polymer, right? Polysaccharide obtained from different seaweeds like red seaweed, gelidium, and also from Selaginella, and different seaweeds are there. From any of these things, you can get this particular agarose powder. And then in the case of electrophoresis, separation of molecules is done in the electrical field. So in this particular one, in the original technique, if you see, the separation of molecules is based on both charge and size, both charge and mass. Actually, it is the charge to mass ratio, but in our experiments, we are directly using the DNA. DNA is always negatively charged. DNA is always negatively charged, whether it is a small DNA molecule, large DNA molecule, whatever it is, the charge is always negatively charged. That's why if you are talking with respect to DNA molecule, separation is based on only the size, means mass. Size means mass. So separation of DNA molecules is based only on the size. Why you are saying like this? Because all the DNA molecules are negatively charged. Always they are moving towards positively charged electrode, that is the anode only. Yes or no? This is what the point. But in simple, without talking about DNA, if they are talking that in the case of gel electrophoresis, separation of molecules is based on, then you need to write both charge and mass. If they are directly talking for DNA molecule, then you need to write size only. That means mass only. That's it. Don't make mistakes here. Many students are making mistakes in this concept. So please observe this very carefully. Anyhow, in this experiment, we are using ethidium bromide, which is a DNA intercalating agent. So with the help of this, so after this attachment, of ethidium bromide with DNA, if it is exposed to UV radiation at the wavelength of 285 nanometer, so then we can see bright orange color band. So these are very important in the experiment. And finally we will talk about gel elution also. Once again quickly I will tell you this. Agarose powder we are purchasing from the market, and initially when this powder is added in the buffer, so it will be like this, it is not completely solubilized. So for the complete solubilization we should boil this. Upon boiling it will become a clear solution, and this agarose when it is cooled to less than 40 degrees centigrade, now it will become a gel. This is the gel. So we are pouring and this is how it will become the gel. And please remember, that's why when the temperature is not less than 40, that means it should be slightly above 40. At that time we are adding the ethylene bromide. Generally in our lab, so we use it to do experiment like this only. We use it to add ethylene bromide. So before pouring in this particular gel, so we will add the ethylene bromide, mix properly so that in the entire gel ethylene bromide will be present. So we are pouring in this one, and this is the comb, and this is having these teeth, so that we are keeping here, and then we are pouring this particular one. Make sure that there are no bubbles here, okay? And then you just leave it for some time. When the temperature is reduced to less than 40, within few minutes, now it will become a solid gel like this. And then take out the comb so that you can see some wells here. And inside this particular gel, if you see, actually this agarose is making some interlinks like this. So that inside this actually there are some pores. You cannot see the pores with our eyes, but actually inside this it is having the pores. So DNA molecules are passing through these particular pores. Understood or not? In this way, it is acting like a sieve. So sieving effect is given by this particular agarose molecule. So because of this particular sieve, what happens? Small molecules can quickly move through this, but large molecules are slowly they are moving through this particular sieving effect given by this particular agarose gel. Understood or not? This is some general information that you need to understand. And after this particular gel we are keeping in this electrophoretic unit, and here we are pouring the buffer for the electricity passes buffer, and some ions are required, so we are using the buffer here. And then we are loading the DNA sample. After loading this particular one, we are connecting with the electrophoretic unit with the power supply, and then switch it on. Then what happens? Now electricity will be supplied here. Now what happens? Now this DNA molecules, since it is having the negative charge, this is about the loading process and all, since it is having the negative charge, it is always moving towards the positive charge only. It is always moving towards the positive charge. Positive electrode is called as anode. So this is always moving towards the anode only. But in this process what happens? Why you are getting different bands at different heights? Because small molecules are moving fast, whereas large molecules are moving slowly. So the molecule that is present far away from the well, this is called well, right? Far away from the well is the smallest molecule. It is present close to the anode. It is present, smallest molecule is present far away from the cathode. Anything they can say. First, now we will talk about the largest molecule. Largest molecule is close to the well. It is close to the cathode, and it is far away from anode. Any of these statements they can ask in examination. So observe very carefully. In this way, based on the size, these molecules are getting separated in the electrophoresis. So once this electrophoresis is completed, we are taking the gel out and we are keeping it on UV illuminator at the wavelength of 285. So it is already preset. You just switch it on. So now the 285 nanometer UV light is coming from the bottom. It is passing through the gel. And here you can see, actually you will see the bands like this. Originally you will see the bands like this. So in this way you are observing this bright orange color bands. Wherever this band is there, it indicates the presence of DNA. Please remember, DNA interacted with the ethylene bromide, that only will be forming like this. Okay. So if DNA is not there, only EDTA cannot give this bright orange color. So in this way, we can identify the location of the DNA bands. Understood or not? With this agarose gel electrophoresis is completed. Please remember. And now we are talking about the gel elution. We are talking about the gel elution. Before going to this is what the NCERT diagram is. So this is the undigested sample. So it is having the complete DNA and it is partially digested. So some bands are there. It is a little bit more digested. So more bands are there. It is completely digested. So many bands, maximum number of bands we are observing here. So in this way, agarose gel electrophoresis is also used to know whether digestion of DNA has happened or not, or if it is happening, at what stage it is present. All these things we can easily know with the help of this agarose gel electrophoresis. Clear? This is what the diagram is. Now gel elution. Please remember, many students are thinking that gel elution is also part of agarose gel electrophoresis. No. So when you are observed here, so with this agarose gel electrophoresis is completed. And now this gel elution is an optional step. Suppose you are interested in isolating this particular DNA molecule. Now let us see that you want to isolate DNA from this piece of gel. Then what you will do? You will cut this piece of gel from that particular one, and then this piece of gel you need to take it in a tube, and then we are adding some solutions so that now it will be solubilized. Once it is solubilized, then we are filtering this one. We are doing this centrifugation, and here we are using some specific membranes, so that now what happens, the DNA will be attached to the membrane and the remaining liquid will be separated out, and now this tube is kept in a separate tube, and now elution buffer will be added here. Elution buffer will be added here, so that now the elution buffer will take DNA along with this. Now this is the elution buffer having the DNA. So in this way, from a piece of gel also we can take this DNA. So in this way, taking DNA from that gel is called as elution. This is optional. It is not compulsory for every time. Please understand this. It is not compulsory every time. So that's why this gel elution is a separate one that you can do after completion of agarose gel electrophoresis if needed. This is what some important information regarding gel elution. Clear? Next we are talking about agarose gel electrophoresis. In a separate paragraph they give agarose gel electrophoresis is employed, means it is used to check the progression of restriction enzyme digestion. So let us take a tube. In that tube we have taken the DNA and we have added the restriction enzyme and we have added the buffer. We kept at the optimal temperature, and now it has to start digestion. Do you think that immediately after addition entire DNA will be broken? No. For the enzyme action it will take some time, right? So suppose you have added and immediately you took some sample and you have observed it is not yet digested, and after 1 hour it is making some bands, after 2 hours it is making more bands, and after 5 hours, suppose let us imagine that after 5 hours complete digestion will be done. So after 5 hours when you loaded the sample, see all the bands were formed. So in this way, what is the level of digestion? So how far the restriction enzyme digestion completed? So that also you can observe with the help of agarose gel electrophoresis. This is the sentence, this is the diagram. This is the diagram. This diagram actually is related to this particular sentence. So in this way, interlink these points also. And now we are talking about the PCR, polymerase chain reaction, and this was actually given by initially invented by this Carey Mullis. Okay, and here actually this particular PCR is also considered as in vitro replication of DNA, in vitro cloning. So here also multiple identical copies are produced, but where? Inside a tube. That's why it is in vitro DNA replication. Clear? And here for this particular process, we need an instrument that is called as thermal cycler. Instrument is called as a thermal cycler. Thermal means temperature, cycler means some cycles of temperatures we are actually maintaining here. So that's why it is called as thermal cycler. And here whatever is the piece of DNA we want to amplify, so for that one we need DNA template, and primers are also required, forward primer, reverse primer, both of them are required. But the important point here is, in the case of DNA replication inside the organism, whether E. coli or human, whatever it is, the primers produced are initially RNA. Later on they are replaced with DNA. But here in the PCR, it is in vitro process. If you use RNA first, who will remove that RNA? Again we need to remove. So why to make it double? So that's why directly we are using DNA primers. Very, very important point. The primers that are used in the PCR process are directly DNA primers. Please remember this. But the primers that are produced inside the organism are RNA primers. This is one important point that you need to notice. And DNA polymerase, Taq DNA polymerase. So this DNA polymerase, please remember here we are going to a temperature of approximately 94-95 degrees centigrade. If you are using the normal DNA polymerase from E. coli or a human, so they will be denatured after 40 degrees centigrade. They are not useful at 95. So that's why we need to use the enzyme, DNA polymerase enzyme, that should withstand 95 degrees centigrade. That should be active even after exposure to 95 degrees centigrade also. That means we need to isolate the enzymes like DNA polymerase from thermophilic organisms, because they are naturally living at very high temperature. So 95 is not high for them. So that's why we are isolating this DNA polymerase from thermophilic bacteria. Different thermophilic bacteria are there. In the NCERT they have given one example, that is Taq DNA polymerase, Thermus aquaticus DNA polymerase. From the Thermus T aquaticus AQ we have taken. So TAQ, Taq, Thermus aquaticus DNA polymerase generally we are using in our experiment. Next one, Mg+2 enzyme divalent cation is required for the activity of these polymerases, and DNTPs for the DNA synthesis. DNTPs are required. Buffer for the maintenance of the conditions. Buffer is required, and even water is also required. And these are the various requirements. And all these things we are adding in a particular tube. Please remember that DNA polymerase used is Taq DNA polymerase isolated from Thermus aquaticus, which is a thermophilic bacterium. Please understand the logic and all. And here in the PCR, total 3 steps are present in each cycle. Each cycle is having total 3 steps. First step is denaturation, second step is annealing, third step is the extension. So during this denaturation, actually the tube is heated to approximately 94-95 degrees centigrade. So at this temperature, what happens? The two parental template DNA strands will be separated because of the heat exposure. And the separation of two DNA strands upon exposure to heat is called as denaturation. That's what. And after this, what happens? Again, we are bringing down the temperature. And here what happens, when you are bringing down the temperature, there is a tendency that parental strands can attach to each other. But we are adding the primers which are complementary to the targeted regions. And these primers are high in concentration. So since primer concentration is high, first primers will go and bind to the opposite regions, complementary regions. So this attachment of primers to the complementary regions is called as annealing. You can also call it as a renaturation also. And after this annealing, what happens? Now this primer should be extended. Who will extend this one? It is the Taq DNA polymerase. It is the Taq DNA polymerase with the help of this DNTPase. It is actually extending in 5' to 3' direction. So that's why this step is called as extension. With this, one cycle is completed. Now again the next cycle, next cycle, next cycle, next. So in the range of 25 to 35 cycles, we are performing these things, so that billions of copies of that particular targeted DNA can be produced. In this way, within a very less time, we can produce billions of copies, billions of exactly same copies for the DNA. So that's why it is called as in vitro DNA replication or in vitro cloning. Understood or not? That's what we are doing. And this piece of DNA we are getting in the billions of copies. Again, we can use it for various purposes. That's it. This is what the aim is. So in the cycle 1, this is what happening exactly. In the regular classes with the diagrams also, everything I have given very clearly. Cycle 2, this is the cycle 3, this is the cycle 4, this is what. And now this is the conclusion. This is most important. Please observe carefully. So after so and so number of cycles, total how many DNA molecules are produced? If they ask, you need to write 2 to the power n. N is the number of cycles. If they are asking that after 5 cycles, total how many DNA molecules are produced? 2 to the power 5. That means 32 molecules are produced. See, cycle number here, total copies here, target copies here. Everything was given here. After 5 cycles, 2 to the power n means 2 to the power 5. 2 to the power 5 means what? Total 32 DNA molecules are produced. Out of 32 DNA molecules, our targeted DNA molecules, how many? What is the formula for this? 2 to the power n whole minus 2n. That means 2 to the power 5. 2 to the power 5 is 32 minus 2 into 5 is 10. So 32 minus 10 is 22. Observe this carefully. This is what the formula is. This is what the formula is. Even many teachers are giving wrong formula for these things. So please observe carefully. For the total DNA molecules, 2 to the power n. For the target DNA molecules, 2 to the power n whole minus 2n. Understood or not? So this is what the formula. At least try to remember the formula. If they're asking that after so and so cycle, how many target molecules are produced, use the formula, you will get the answer. And here observe the table carefully. For the targeted molecules, actually targeted molecules synthesis is starting in which cycle? From the third cycle onwards, actually the synthesis is starting. In the first and second cycles, targeted molecules are not produced, but still from the third one they are forming. And after few cycles, what happens? When compared with this one, anyhow, these are the total number always higher than targeted copies. But if you are observing about targeted and non-targeted copies, slowly, targeted number is crossing that non-targeted cycle. Actually, this targeted copies number is increasing in geometric progression, whereas non-targeted DNA molecules number is increasing in arithmetic progression. That's why definitely this number will be much higher when compared with non-targeted DNA molecules. Is it clear? These are some important points that you need to remember here. Now this is one application type, RT-PCR. What is RT-PCR? If you clearly observe, for the COVID-19, for the coronavirus confirmation, the confirmatory test is RT-PCR. Please remember, you know PCR, polymerase chain reaction. But what is this RT? This RT is reverse transcription. Please observe carefully. This RT is reverse transcription. Why you are going for reverse transcription for COVID-19? So you need to understand the logic there. Please observe carefully. Actually, in the case of PCR, from the DNA you are producing DNA. Please observe this. In the case of PCR, template is DNA. Formed molecule is also DNA. But in the case of COVID-19, the disease is caused by novel coronavirus NCOV2, and that virus has RNA as genetic material. Please observe carefully. That is not a retrovirus, it is a RNA, simply RNA containing virus. And that virus, if you want to do PCR, can you use that RNA as it is for PCR amplification? No. First you need to isolate RNA from the virus. Once you are getting this RNA, for the PCR, what is initially required? DNA is required. So this viral RNA should be converted into DNA. How you will convert RNA into DNA? By reverse transcription. So that's why first you are doing reverse transcription for the conversion of RNA into DNA. Once DNA is formed, then again you can proceed with normal PCR and use the primers specific to viral DNA. In this way we are preparing this. That's why RT-PCR we are using. First reverse transcription you are doing so that RNA is converted into DNA, then we are doing the regular PCR. This is what the process is. So if ultimately if you are observing the amplification, that means that the patient is having that particular viral infection. Suppose you did not get the amplification, that means that patient is not having the infection. That's it. In this way we can give the confirmation with respect to this COVID-19. Clear? Next one. Isolation of DNA, genomic DNA isolation. How exactly we are isolating DNA? But the thing is, why you need to isolate DNA? In this DNA, our target DNA is present. Our gene of interest is present. To get that gene of interest, first you need to isolate the complete DNA. For the isolation of complete DNA, first you need to break the cell. For breaking of the cells, we already studied some lysis enzymes. For the bacterial cell wall degradation, it is lysozyme. For the plant cell wall, cellulase. For the fungal cell wall, it is chitinase. In this way, various enzymes we are using. After that, we are using various chemicals so that we are breaking the cell so that all the contents will come out. So in the cellular content, what are present? DNA is present, RNA is present, lipids are present, proteins are present, carbohydrates are present. Use carbohydrates so that carbohydrates are broken. Use lipases so that lipids are broken. Use proteases so that proteins are digested. So use RNA so that RNA will be digested. Or digest all these things. So what is remaining? DNA is remaining. Isolate the DNA. That's it. In this way. So during this isolation, what actually we are doing is, initially in this step what actually happens is, this DNA is present like some threads that are floating in this particular fluid. So these threads we can actually collect with the help of a glass rod. So you can spin it like this so that those threads will coil around this particular glass rod and then take it out. So in this way, so in this way, this is actually called as a spool. So with the help of spooling, so since you are coiling like this, suppose this is the glass rod, DNA threads will be coiled around this one so that you can take it out like this. So in this way, collecting DNA like this is called as a spooling. So with the help of this spooling, we can collect this DNA in a separate tube. And after that we are adding the ethanol, absolute alcohol. It is also called ethanol, means absolute alcohol we are adding, so that what happens? Now it will cause the precipitation. So after precipitation, so we will do the centrifugation and we will remove the alcohol. And now this precipitated DNA, once again we will dissolve in elution buffer. Okay, or solubilization buffer will be there. So in that solubilization buffer, you can easily solubilize the DNA, and that DNA you can use it for further experiments. So in this way, we are actually isolating the DNA from the particular cell. Clear? Then we are talking about the process of recombinant DNA technology. In the NCERT, very clearly they have given all these steps one by one. One by one they have very clearly given. Observe this. First we need to isolate the DNA, and then fragmentation of DNA by restriction endonucleases, and isolation of the desired DNA fragments, and the ligation of DNA fragment into a vector, then transferring recombinant DNA into the host, and then culturing the host cells in a medium at the larger scale, and then extraction of the desired product. This is the sequential steps, and these sequential steps you need to remember in the same sequence only. Sometimes randomly they will give this and they will ask you to arrange them. So you need to remember this particular sequence. And now here I am telling you the process of selection with the help of this PBR 322. If you are using PBR 322, in the NCERT they said that if gene of interest is introduced at the BAM-H1 site, then how you will do the selection. I will try to explain the entire process a little bit quickly, so observe very carefully. On one side we are taking the gene of interest, I am writing GOI, gene of interest. On the other side we are taking PBR 322, this is the PBR 322 vector. And here it is having ampicillin resistant gene, and also it is having tetracycline resistant gene. And within this tetracycline resistant gene, it is having the BAMH1 restriction endonuclease site. Clear? In the NCERT, they have given the explanation with respect to BAMH1. So that's why I am also giving the explanation with respect to BAMH1. So here what is the point is, this gene of interest should be introduced into the PBR322 at BAMH1 site. So how you will introduce this at BAMH1 site? So now you need to cut with BAMH1 restriction endonuclease. So please remember, here if you want to introduce, now here also we need to cut with BAMH1. Very important point, so please observe carefully. So wherever we want to introduce, the corresponding enzyme we need to use. So if you are cutting the plasmid with this particular enzyme, gene of interest also should be cut with the same enzyme. What is the advantage with this? It will produce the sticky ends, and these sticky ends are complementary here also. So now till now it is circular molecule, but now it will become linear. Now it will become linear here. So observe very carefully here. It is becoming linear here. But which one is broken here? Tetracycline resistant gene is broken here, but ampicillin resistant gene is normal here. Understood or not? And here, this is the gene of interest that is having the sequences that are complementary to this particular sequences, because it is also cut with BAMH1. And now what we are doing? We are combining both of them in a single tube, and we are adding the enzyme called as a ligase. And ligase enzyme we are adding, and we are mixing in this tube. So in this tube, what are the various combinations that we are observing? Now observe very carefully. What actually we are observing here? So here what happens is, now this is the ampicillin resistant gene and this is the tetracycline resistant gene. So first and foremost important option is, since ligase we are adding, and since these two are complementary to each other, it can attach with each other. So that again they are forming the self-ligated plasmid. It is the self-ligated plasmid. That means once again the original plasmid was formed here. And what is the other option? This piece of DNA can be attached here. So in this way, here it will form recombinant DNA. So ampicillin resistant is normal, but here I am making like this. So from here to here it is tetracycline resistant gene, but tetracycline resistant gene got broken, and in between this gene of interest was introduced. That means here tetracycline resistance is not present now because the gene is broken. Now it cannot offer resistance to tetracycline. So in this way, this is self-ligated plasmid, and this is the recombinant DNA, and it is also called as a chimeric DNA. It is called as chimeric DNA. Understood or not? This is the recombinant DNA. In this way, we made the recombinant DNA. In a separate tube, we are taking the competent host. Who is acting as competent host? It is the E. coli, chemically treated, calcium chloride treated E. coli. That's why it is the competent host. It is ready to take external DNA. So now these two we are combining in a single tube. And once we are combining all these things in a single tube, and then initially kept at 4 degrees centigrade, then we are giving the heat shock, and then again keeping it 4 degrees centigrade, and this process is called as transformation. This process is called as transformation. So after this step, we will get various possibilities. What are all these possibilities? Observe very carefully. Even after this particular step also, some E. coli do not receive any external DNA. So they are present like this only. So okay, names I will give you separately. Next one. Whereas some E. coli will take this self-ligated DNA. Some E. coli will take self-ligated. Self-ligated means it is having ampicillin resistance and tetracycline resistance. Understood or not? Whereas some E. coli are receiving recombinant DNA, which is having only ampicillin resistance. There is no tetracycline resistance. In this way, three possibilities are there. Some bacteria do not receive anything, are called as non-transformants, whereas some bacteria which are receiving something from the outside, something they have received, these are called transformants. And within the transformants again, the one that received the recombinant DNA is called as recombinant. The one that did not, the one that received self-ligated plasmid is called as non-recombinant. These are the names that they have given very clearly in NCERT. You need to know this. And all these are present in the single tube. By simply looking at this, you cannot identify recombinants. So we need to go for the selection process. So in the selection process, what we are doing is, first we are taking the plate, agarose plate, agar plate, agar nutrient plate. This is the nutrient agar plate. And in this plate, what actually we are doing is, see here, what is our aim? We need to get this particular recombinants. So what is common for both the transformants? Ampicillin resistance is common. So since they are resistant to ampicillin, in the medium we are adding ampicillin antibiotic. Once we are adding ampicillin in the medium and this entire content we are spreading on this plate, then what happens? First thing that happens is these bacteria will die. Why they will die? Because non-transformants do not have resistance to ampicillin. If they don't have resistance to ampicillin, in presence of ampicillin they will die. So that's why these will die. Done. Now who will grow? Now these two will grow. Now these two will grow. Now let us see. We don't know which one is which one, we don't know. Simply they are growing like this. They are making hundreds of colonies. For understanding, I gave just 8 colonies, but hundreds of colonies will be formed here. Now from this, now here both of these are there. And then further one. So please remember, in this particular one, between non-transformant and transformant, which one we have used? Ampicillin resistance we have used as selectable marker. To distinguish between non-transformant and transformant, we used ampicillin resistant gene as selectable marker. And now to distinguish between non-recombinant and recombinant, we need to use tetracycline resistance. And for this, we need to go for replica plating method. We need to go for replica plating method. In the replica plating, exactly same size plate we need to take. Exactly same size plate we need to take. And this is also agar plate, nutrient agar plate, but this time in the medium, instead of ampicillin, what we need to add? Tetracycline we need to add. Here we are adding tetracycline. Here we are adding tetracycline. And now you need to transfer all this. Can you transfer one by one here? No. Hundreds of colonies are there. So what you need to do? Now here impression type technique we need to take. So what we will do is, for this plate, the matching exactly same size, like round stamp like structure we are taking, and below that we are keeping velvet cloth, and gently press on this. Don't press completely. So gently press on this, so that what happens, exactly at the locations, exactly at these locations, cells, majority cells are present here, but some bacteria they will attach on the cloth. And now by matching this particular point, we are transferring the bacteria here. Simply press on this, so that bacteria are transferred here and allow them for the growth. Now what happens is, in this particular one, some bacteria are growing here. Some bacteria are growing here, and some bacteria died. Now here there is no growth, they died. And only some are growing here. That's it. And now you please remember, here you don't know. Here you don't know which one is recombinant, which one is non-recombinant. But here, can you identify which one is recombinant, which one is non-recombinant? Yes. Who can grow on tetracycline? Is it recombinant or non-recombinant? Please remember, it is the non-recombinant which is having tetracycline resistance. So, on this plate, who is growing? It is the non-recombinant. Non-recombinant is growing here. Non-recombinant is growing. But actually, we want recombinants, and these recombinants are dead here. They are killed here. Then what is the use? Even though recombinants are dead here, there is a use here. What you need to do? Take both the plates. And this is the point for matching, right? Now compare here. Now here the colony, this colony will match with this. That means this is non-recombinant. This colony will match with this. This is non-recombinant. This colony will match with this. This is non-recombinant. This colony is matching with this is non-recombinant. Understood or not? So start comparing both the plates. So the locations of this particular colonies on this plate, wherever they are matching here, all these are non-recombinants. And here the remaining. What is this remaining? Here there are no colonies, but here there are colonies. So what will be these colonies will be? By default, these colonies will be recombinant. Understood or not? Here non-recombinants will grow, and the required recombinants they are dying here. They cannot grow here. Even though they don't grow here, by comparing with this, on this master plate, you need to identify which one is recombinant, which one is non-recombinant by comparison with this. In this way, you are identifying the recombinants. So once you identify the recombinant colonies, take some cells and then you just grow in the medium and then let them grow. That's it. This is how you are actually identifying these particular recombinants. And this is the selection of recombinants using PBR 322. This is what actually we are doing here. Is it clear? This is what the selection based on PBR 322. Very, very important thing. Don't make any mistake. This is what. Okay. Next one. So here I have given the different questions. You need to understand. If gene of interest is introduced at BAM-H1, then recombinants, can they grow in ampicillin medium? Can they grow in tetracycline medium? Non-recombinants, can they grow in ampicillin medium? Can they grow in tetracycline medium? Then What is the selectable marker to distinguish between transformant and non-transformant? What is the selectable marker to distinguish between recombinant and non-recombinant? These are all the questions with respect to BAM H1 site. Now another question. If gene of interest is introduced at SOL1, then what are the results? If gene of interest is introduced at PVU1, if it is introduced at PST1, if it is introduced at other locations, each and everything is a separate question. Now each and everything is a separate question. Now you can understand how many questions can be formed on this. So for all these things, the basic thing is you need to understand the procedure. How exactly we are performing the procedure, how exactly we are doing the selection. So if you understand that, all these things you can do without using pen and paper. Clear? So this is what the level of accuracy that you need to understand and that you need to practice. These all will come only by the practice. So please start practicing this. And with that, selection based on PBR 322 is completed. Now we are going with selection based on PUC 18 or 19. And here what are selectable markers? Ampicillin resistant gene and LAGZ. And here this is about the blue and white colony screening, and not clearly given in NCRT, but at least they mentioned. So you need to understand this. Now let us see. Here also the same process. Here also the same initial process is same only. Here also, suppose this is the gene of interest. This is the gene of interest, and this is the PUC 18 or PUC 19. Anything you can take. And here it is having ampicillin resistant gene, and it is having LAGZ gene. Please remember here, LAGZ gene. And here you need to cut with a restriction endonuclease. You need to cut with a restriction endonuclease. Please remember, we want to introduce within LAGZ. Cut with same restriction. So here you will get the sticky ends. You will get the sticky ends. This is the gene of interest. And now this plasmid will become linear here. Linear here. Yes or no? LAGZ got broken, but ampicillin resistant gene is normal. Yes or no? And then combine both of them using the ligase enzyme. And here you will get two options. First one is self-ligated. Once again it will become ligated, so that you will get again ampicillin resistance and LAGZ. And what is the next option? This is introduced here, so that recombinant DNA is produced. But this recombinant DNA is having only ampicillin resistance. LAGZ is lost. Clear? Next one. And then in one tube they are present like this. In the other tube we are taking the E. coli. Please remember, for PUC 19 also, E. coli is the host. We are taking the host. Combine both of them. Transformation. Transformation means initially keep it at mix, and then keep it at 4 degree, then 42 degrees centigrade, and again at 4 degrees centigrade. And then what are the options that we are getting here? Observe carefully here. Here what happens is, some bacteria do not receive anything. This is called as non-transformant. Some bacteria receive self-ligated plasmid. That means ampicillin resistance and LAGZ are present. This is transformant. Within the transformant, what type it is? Non-recombinant. Yes or no? Next one. Some bacteria are receiving recombinant DNA. They are having only ampicillin resistance, because other side they are having gene of interest. So this is also a transformant, but this is recombinant type. And how exactly we will select here? Here also you are taking the plate, but this time single plate is enough. This is the advantage. In the PBR 322 you need to make 2 plates, but here single plate is enough. And here it is the agar plate again, and this time what you need to add with respect to ampicillin resistance. So that's why first add ampicillin in the plate. Once you are adding ampicillin in the plate, what happens? Now these will die, non-transformants will die, and now here both of these will grow. Understood or not? Growing. And now to identify these two things, again use the other one, LAGZ. What is the concept of LAGZ? Now observe carefully. If LAGZ is present, it will produce beta galactosidase. And please remember, in the molecular basis of inheritance, under lacopran also we studied the same thing. But there we are using lactose as substrate. But here we are not using lactose, because lactose is colorless. Upon breakdown, it will produce glucose galactose. They are also colorless. So that is not useful here. And here we are using a different substrate. This substrate is called as XGAL. XGAL is a colorless substrate. And it is also called, many students are making mistake here, so observe carefully. It is also called as chromogenic substrate. Observe carefully. What is the meaning of chromogenic? I am saying colorless. Chroma means color. But how it is chromogenic? Observe carefully. Meaning, chroma means color, genic means producing. So this is a color producing substance. substrate, it is not having color. It is a color producing because the product is a colored product. Product is a colored product. It is a color producing substrate. So that's why it is the chromogenic substrate. So what is the color of this chromogenic substrate? It is colorless. But what is the color of this particular product? In this case, when this beta X gal is broken down, it will produce blue color product. So please observe carefully. Who will give this blue color? Those which are having lag Z. So if lag Z is present, it will become blue color colony. So understand the concept here. First we will see this. I will use this blue color here. Okay. Now let us see. Who is having this lag Z? Non-recombinant is having lag Z. Suppose this is the blue color colony. Suppose this is the blue color colony. Suppose this is the blue color colony. So who will form the blue color colony? It is the non-recombinant. So blue color colonies are non-recombinants. Blue color colonies are non-recombinants, whereas white color colonies, these are the white colonies because they don't have lag Z. If lag Z is not there, even though X gal is added to medium, you need to add X gal to medium so that non-recombinants can convert X gal into blue color product. So that colony will become blue. Yes or no? Colony will become blue. And here these are the white color. So that's why they are recombinants. So originally colony is white in color. You need to understand this. From where you are getting this white color? You are saying that colorless substrate is colorless. So please understand this. Originally, when E. coli colonies, when E. coli is growing on agarose plate, it will form naturally white color colonies. Naturally, it is forming white color colonies. But here X gal is present. These are having lag Z, so that they are forming the blue color colonies. That is what the difference is. Okay. The blue is actually this type of blue. The blue is actually this type of blue you are observing in the original colonies. So remember this. So in this way, in this particular one, blue color colonies are non-recombinants. These are non-recombinants. Blue color colonies are non-recombinants, whereas white color colonies are recombinants. So in the single plate, simply by looking at the color, you can identify that, yes, these are the recombinants. So take some amount of this and then grow them. That's it. In this way, you are doing the selection of these particular recombinants. That's it. Understood or not? This is what it is. So blue color colonies are non-recombinants, white color colonies are recombinant bacteria. That's it. So in this way, we are actually identifying this. So in this way, with the help of some vectors, we are actually transferring gene of interest into the targeted cells. This is what actually we are doing. Now here we are studying without the vectors. Even without any vector also, directly gene of interest can be transferred into host. That's why these are direct gene transfer techniques. Here vector is not needed. What is this? Micro injection. Generally it is used for the animal cells. So here you are holding the animal cell with the help of injection. So inside this, what actually you are doing? Gene of interest. Directly it is introduced into the nucleus of this particular cell. And there with using some mechanisms, it will become part of the chromosome. That's it, done. This is called as micro injection. And other one is called as biolistic or gene gun method, and this is most useful for the plant cells because they are having cell wall, they can resist some force. So that's why here what we are doing is we are taking some tungsten or gold particles. On this we are giving the targeted DNA as a coat, and now they are taken in the gene gun. So now it will be facing this particular plant cells, and then you just trigger this, and now they will go with high velocity and they will enter into the plant cell. And now once they are entering into this plant cell, now this coated target DNA will come out of it and it will go and become part of plant chromosome. That's it. So in these techniques, vector is not used. That's why these are direct gene transfer techniques. That's what we are talking. And whatever it is, with the help of vector, without vector, whatever it is, you have transferred gene into the host. Once it is introduced into the host, now our aim is to express them. So gene is originally from one species and we are expressing this in a different species. And now this host which is carrying gene from some other species is called heterologous host. And when it is producing that protein, it is called as a recombinant protein. So in this way, it is producing recombinant protein. But initially, before directly going with large scale, first we need to do in the laboratory at a small scale. And once we are optimizing all the conditions, then we are going with a pilot culture, and there we will decide whether it is profitable for us or not. If it is profitable, then we will go with large scale production in the bioreactors. So bioreactors are large tanks where we are converting some raw materials into some different products or enzymes or some other bioactive molecules. Different things can be produced here, and for that purposes we are using some enzymes, we are using some cells, some animal cells or plant cells, sometimes we are using some microbes like bacteria, yeast, many things we are using. This is what actually we are doing in the case of this bioreactor. And if we see this bioreactor structure in simple, see actually in the microbes in human welfare, they have given these tanks, no? They are actually bioreactors. And this is a rough diagram. So these are the large vessels, generally they are cylindrical. At the bottom they may be curved shaped or they may be flat. And inside this generally, in the liquid culture if you are talking, so here we are actually pouring some liquid, and that liquid medium is called as culture broth. And if the organism that is growing here is aerobic, then we are actually mixing the contents with the help of this impeller blades and all. And if it is anaerobic, then we are not mixing this. Understood or not? Aerobic, anaerobic, anything you can grow. If it is aerobic, two things are important. One is mixing, and other one is regarding the sparger. Sterile air is supplied with the help of a sparger. Sparger. If sparger is present, it is called sparged. If stirring is also there, stirred. So sparged, stirred tank bioreactor. So based on what are present, we are giving the name. If these are not there, it is called a simple bioreactor. That's it. In the simple bioreactor, stirring is there, but sterile air will not be supplied here. So in this way, we are using all these things, and to maintain these optimal conditions, some separate ports for the pH, for the acid base balance, all these things will be there. And once this entire content is removed, we need to wash this and we need to clean this, and for that we are actually providing the steam here. So steam is having very high heat and temperature, so that it can helpful for sterilization on inside, so that the previous content will be entirely sterilized, and after removal, if any microbes are left over, they will be killed, so that it will be sterilized, so that again we can use it for the next batch. Like this we can use it. So not only this, when we are mixing this nutrient broth, there will be some foam that is formed at the top, and this foam may prevent the re-dissolution of this particular gas. So that's why foam breakers are used. If the foam breakers are removing the foam, what happens? The air supplied from the bottom in the form of bubbles, because of its more surface area, now it will be easily diffused into the medium, and the remaining air is coming to the top, and again it is going back into the medium, so that again it will be dissolved, so that aerobic organisms, they can easily use this particular oxygen for their growth. This is some important point that you need to focus. Clear? These are some things that they are talking here. So in this way we are growing these particular things, and finally they will release the product into the medium. And please remember, here different things are there. Some are called as batch cultures. You are adding all the contents and maintaining conditions. Once product is formed, entire contents you are taking out, that is called as batch. In the fed batch, intermittently you are adding substrate, that's it. And after some time, at once you are taking all the contents, that is called fed batch. In the continuous cultures, what happens is, continuously at regular intervals you are adding the substrate, and at regular intervals you are collecting the product. So in this way it is a continuous process. The batch is running continuously. That's why it is called as continuous culture. What is the advantage with continuous cultures? Because of this continuous cultures, always the cells are in most exponential, most active phase, that is called exponential or logarithmic phase they are present. So that's why they can produce maximum quantity of the product. That is what the advantage is. Clear? Next one, aerobic organisms. For the maintenance of them, we need to maintain aerobic conditions. If they are anaerobic, don't provide oxygen, don't mix the contents, so that anaerobic bacteria can be used. Initially these bioreactors were used for growth of these particular anaerobic things, that's why they were called as fermenter. But today also we are using the term fermenter, but still we are using some aerobic organisms also. That's why it is a general term. Okay, next one, what is downstream processing? Once this particular product is produced in the medium, whether it may be present inside the cell or it may be released into the medium, based on the experimental procedures it will be dependent. So once that particular protein is produced, after that whatever you are doing, all the things will come under downstream processing. After the production of the recombinant protein, whatever we are doing, all the things will come under downstream processing. So in the downstream processing, what are the simple things? First we need to isolate the particular product from the remaining contents, and then you need to purify them. So all these things will come under this particular downstream processing, again based on the type of material. If suppose if you want to make some quality tests, so then we need to perform some quality tests before marketing, and sometimes clinical trials may be required based on the type of the product molecule, and sometimes for the long term storage even we are adding some preservatives also. So in this way we are actually doing all these things after this production of the recombinant DNA protein. So that's why all these things will come under the downstream processing. and these are the different restriction nucleases and from which organism it was isolated, simply for understanding purpose only, because these names were mentioned in that PBR 322 vector map. So that's why I have given. Whatever you can remember, but at least compulsory is EcoR1. EcoR1 is compulsory. Okay, next one. Regarding the reporter gene, in the exercise they have mentioned about this. What is the reporter gene? What is a news reporter will do? See, we are sitting in our home and news reporter is actually reporting something that is happening there. So simply by sitting in our home, we are able to see what is happening there. Same logic apply here. Reporter genes are there. There are some genes which are used as reporter genes. What they will do? Suppose I am a scientist, so I want to clone some gene and I want to get some product from this gene. But directly I cannot see this particular gene product. So I will add a particular reporter gene. Once I am adding this reporter gene, what this reporter gene will do? When this gene is expressed, this is also expressed. And if I can able to detect this particular product, it means that presence of this particular product means that this product is also present, because this will be produced only when it is produced. So in this way, reporter genes are the genes that are introduced along with the gene of interest to ultimately monitor or to know whether that gene is expressed or not. So in this way, there are different genes that are present, and one simple thing is even you can use this LAGZ gene also as a reporter gene from our syllabus. Not only this, that is green fluorescent protein producing gene and red fluorescent protein producing gene. Likewise, many things are there, and that you will study in higher classes. This is what one important thing that you need to understand. And in this way, and report enzyme examples: chloramphenicol acetyltransferase. So this gene producing this enzyme. So by doing the assay for this particular enzyme, we can easily understand whether it is present or not. Beta galactosidase produced from LAGZ. So that also we can use as a reporter. And luciferase. So here we are observing some illuminance. So that illuminance we will record it. So that also we can use. So different are there. But anyhow, you don't need all this. But second one, beta galactosidase is the protein produced from LAGZ gene. So LAGZ gene can be used as reporter gene. Okay, so this is what one important point that you need to understand. Regarding the terms, already I told about this in the molecular base of inheritance. Transformation term we are using for the bacteria and all the eukaryotic cells except animal cell. Please remember this. Transformation term we are using for all the prokaryotes and eukaryotes except animal cells, because in the animal cells transformation means conversion of normal cell into tumor cell. In the case of animal cell, that word transformation we are using for conversion of normal cell into tumor cell. That's why if you are introducing a gene into the animal cell, don't use the term transformation. You need to use the term called as a transfection. This is what the point is. Regarding transfection, I told you, if the gene DNA or the gene is introduced from the virus into the bacteria, then it is called as transfection. That's it. These are some important terms. With this biotechnology first chapter is completed. Before concluding, once again I am telling you, revision lectures are available both on LivePrep app and also on YouTube channel, but test series can be given only through the app. Clear? So with this part is completed. In a separate lecture, we will meet with a different chapter. Thank you.